Everyone, I am Auntie Danielle, and today we will be reviewing CSEC biology. Our first topic that we will be reviewing is photosynthesis. All right, so now we'll get into our past papers. So the first question, as we can see, says to define the term photosynthesis. Now, when we're defining the term photosynthesis, we're looking for some key terminology to be used. And a sample definition for photosynthesis could be the process by which plants or algae or even some bacteria uses simple inorganic molecules, which is our carbon dioxide and water, in the presence of chlorophyll and sunlight to produce our organic molecules, which is our glucose and oxygen. It's very important not to forget that, yes, our carbon dioxide and our water is our raw materials, but don't forget that this process of photosynthesis is done in the presence of sunlight and chlorophyll. Now, the next question asks us to write a balanced chemical equation for the process of photosynthesis. Before we get to the equation, we realize it's worth 2 marks, which means you will be writing out the equation and you will also be balancing the equation. So at this time, we will now bring up our chemical equation for photosynthesis. All right, so by this time, we will see our equation on the screen, and we will see that we will have 6 molecules of carbon dioxide with 6 molecules of water. We will see an arrow. We normally have our arrow, all right, indicated, and we normally indicate our sunlight over our chlorophyll. And then we will produce our glucose, which is C6H12O6. And we will also have 6 molecules of oxygen being produced as a byproduct. All right, at this time, we'll now move on to our other question, or the remainder of our question. So it says to describe the movement of water from the soil to the photosynthetic cells of the plant. Before we answer this question, we have to talk about what is the photosynthetic cells of the plant. This beautiful terminology, it's just highlighting to you this is where photosynthesis would take place. And normally our photosynthetic cells of the plant is our leaves. So we wanted to describe the movement of the water molecules from the soil to the leaves of the plant. Which means that first we're going to start off with the process of osmosis. Now, I know from a long time we'd all have learned the definition of osmosis, which is when we have our water molecules moving from an area of high water concentration or high water potential to an area of low water concentration across a semipermeable membrane. So in the case of what they're asking us, we'll have the water molecules moving from an area of high water concentration, which would be in the soil surrounding the root, to an area of lower water concentration, which is inside the root hair cells. All right? The root hair cell itself is acting like the semipermeable membrane. Now, this movement of water into the root hair cell will create a gradient which allows the water to move from the root hair cell through the other types of cell like our cortex, endodermal cell, all the way until we reach to our vascular bundle in our root, where we'll have our xylem and our phloem. Normally, we would have root pressure taking place at this point, where we essentially have because of the accumulation of water, we have like a push of the water into the stem. Now, the next process we're going to really define, or the question wanted to define or talk about, is your capillarity, which has in your cohesive and your adhesive forces. Your cohesive forces is talking about when your water molecules will be sticking to each other, while your adhesive forces will be talking about when your water molecules stick to the surface or to the walls of the xylem vessel. Both of these forces work together to help the water molecules to actually climb up the xylem wall, where you will have some water molecules sticking to the wall of the xylem, while you have some water molecules are sticking to the other water molecules. All right? This helps it to actually climb up the wall of the xylem. Now, to continue talking about the movement of the water to our photosynthetic cells, we will talk about transpiration, which involves the evaporation of our water molecule from the leaves of our plants. And to talk about transpiration, we have to talk about our transpiration pull. Now, when we're talking about transpiration pull, we normally talk about the arrangement of our water molecules, where we like to say that each water molecule is essentially connected to another water molecule, where most time when I'm explaining it, I talk about the fact that we have our pearls like connected to each other, like in a bracelet or in a necklace. Now, when we have transpiration taking place where one water molecule is essentially evaporating, it pulls on the other water molecule that it was attached to, which helps to bring it from the stem into the leaves of the plant. Now, the next question is asking you to distinguish between the role of your intercellular airspace and that of your stomatal pore in your leaf in the process of photosynthesis. Now, we all know about our stomata, which is what they're calling the stomatal pore. Your stomata and your intercellular airspace is essentially helping with the process of gaseous exchange. Your stomata is acting like an entry point for carbon dioxide to diffuse into your leaf, and an exit point for your oxygen to diffuse out of your leaf. While your intercellular air spaces tend to be found in your spongy mesophyll layer of your cells, and what it does is act as a gaseous exchange surface area, which helps to increase the rate of gaseous exchange. Okay? Now, the next part of the question is saying, explain why the rate of photosynthesis decreases on a very hot day. Now, when we're talking about photosynthesis, we know that there are different factors that can affect the rate of photosynthesis. And typically, when we're thinking of like something limiting the process of photosynthesis, we talk about our carbon dioxide concentration. And that's because we're typically or normally looking at light intensity. But the question itself says photosynthesis decreases on a very hot day, which is indicating to you that temperature would be the main focus or the main factor it is viewing or focusing on for this question. Now, your normal temperature range or approximate temperature range would be between 35 degrees Celsius to 37 degrees Celsius. On a very hot day, the temperature tends to go above this particular temperature range, which means now that while the temperature will increase once it gets to that maximum or optimum temperature for the enzymes in the leaf, above that optimum temperature, the enzymes will start to denature, and this will decrease the process of photosynthesis. The next part of the question is saying, state one role of chlorophyll in photosynthesis. The main role of chlorophyll in photosynthesis is to absorb our light energy so that this light energy can be used to make food. And it's also used for the splitting of water, but the main role of chlorophyll is to absorb the light energy to make food in the process of photosynthesis. All right, so the last part of our first question says, outline 2 ways in which photosynthesis is important to living things. Now, the two things that we'll be focusing on is stability of ecosystem/food chains and the production of oxygen/reduction of carbon dioxide. Now, for our stability of our ecosystem/food chains, we know that our photosynthetic organisms would normally be our plants, which are our autotrophs or our producers when we're talking about our food chains. And we know that directly and indirectly, all food comes from our producers. So when our producers take this light energy from the sun and carry out the process of photosynthesis, they will not just be providing food for themselves, but they will also be providing food for other organisms. And when these other organisms are able to eat the food, they can use it for their different metabolic activities, and so that we can have a thriving ecosystem to maintain interpersonal relationship and codependence. If it is that we have our producers, our photosynthesis not taking place, then we essentially will have the collapse of our ecosystem. The other point, or the other way photosynthesis is important, as we said, is for the production of carbon dioxide and the production of oxygen and the reduction of carbon dioxide. Now, when photosynthesis takes place, carbon dioxide is normally removed from the atmosphere for the process of photosynthesis. This removal of carbon dioxide from the atmosphere will help to reduce the level of carbon dioxide that is present in the atmosphere, which helps essentially with maintaining the amount of the carbon dioxide level, which helps with maintaining our greenhouse gases for the greenhouse effect. When we focus on the production of oxygen, we know that different organisms, like humans, use oxygen to carry out respiration. And we know that these organisms, by the process of respiration, will produce cellular energy so that we can actually thrive and carry out our different metabolic activities. So those are two ways in which photosynthesis is important. All right, we will now be moving on to the next topic, where we have enzymes and digestion. All right, so for our enzyme and digestion question, the first thing we see the stem of the question, where it is asking the students to investigate the effect of temperature on the rate of reaction for the enzyme catalase. They say that the students will be using crushed potatoes as the source for the enzyme, and they will be placing 5 milligrams of each in 7 test tubes. They are asking them to add 3 centimeters cube of 1% hydrogen peroxide to each test tube and to place them into water baths kept at the specific temperatures listed: 0, 10, 20, 30, 40, 50, and 60 degrees Celsius. Then the students would have been counting the number of bubbles released per minute from each test tube, and they would have recorded the data in the table that we are seeing on screen. Now, the first part of the question is going to ask you to plot a graph with the graph paper or grid provided in your booklet. At this point, we will bring up the graph that we would have plotted based on the data that we have been given. Now, it's a 4 marks question, which means that, as you can see on the graph on screen, there are certain things that you should have on your graph. You should have your title, which we know should be in capital letters and underlined, and it should be a self-explanatory title. Our title says graph showing the number of oxygen bubbles per minute against temperature. You should also have your axis labeled, where you will have your y-axis showing the number of oxygen bubbles per minute and your x-axis showing the temperature and the unit listed in Celsius. You should also have your scale present on the graph, as you can see at the bottom of our graph, where we have on the y-axis 1 centimeters to 1 unit, while on the x-axis 2 centimeters to 10 units. And then you would correctly plot the data which have been given to you in the table provided. Now, before we move on, the second step of the question is asking you to use this same graph to determine what would be the optimum temperature for the enzyme. Now, based on the shape of the graph that you're seeing, you would be able to identify that the optimum temperature would be 40 degrees Celsius, which would have been essentially the peak of the graph or the highest point of the graph, where the most number of oxygen bubbles were released. Now, the next question is asking about the shape of the graph. So there are different stages that it's asking you to describe the shape of the graph. The first stage would have been 0 to 10 degrees Celsius, which essentially would have been the initial shape of the graph. All right? Now, I should highlight that this question is 6 marks, which means that it's 2 marks per section or shape that is identified. Now, for the 0 to 10 degrees Celsius, you would have had a slow rate of reaction, meaning that the collision speed between our substrates and our enzyme would have been low because the temperature would have been low, which means the kinetic energy of these molecules would also have been low. All right? Then we'd have moved on to the other part of the graph, which is 11 to 40 degrees Celsius. Now, we would have had the temperature increasing, which tells us that the rate of collision has also increased or will be increasing as well, because there will be a lot more kinetic energy being supplied or being given to the different molecules. So they will be colliding a lot more with each other. Now, your graph should be increasing or would have been increasing until you reach the 40 degrees Celsius, which we had identified previously as our optimum temperature, which means at that point we would have had maximum rate of collisions, because that would have been the optimum temperature of the enzymes where it works the fastest, or we have the most bubbles being produced. Now, the last part of the question is asking you about the last shape of a graph, which is above 40 degrees Celsius, which would be 40 to 60 degrees Celsius based on the data that we were given. Now, we would have realized that we saw a decline in the graph or a drop in the amount of bubbles produced. This is because even though the temperature is increasing, what we do know also happen is that the temperature affects the enzymes, specifically the active site of the enzymes, and a process that we know to be called denaturing takes place. And denaturing simply means that the active site of the enzyme would have started to change shape, which means the substrate could no longer fit into the active site. And since the substrate can no longer fit into it, our rate of reaction would start to slow down. Now, they would ask you to summarize essentially those main points in this particular question. Now, the last part of it is saying, write a conclusion that the students could draw from the results. All right? A simple conclusion could be that the increase in temperature increases the rate of photosynthesis up to the optimum temperature, where the reaction takes place the fastest. Past your optimum temperature, your rate of reaction will slow down as your enzymes start to denature. All right, now we're moving on to the other parts of our question. We're still on enzymes, and it says, state one reason why living organisms need to maintain a constant internal temperature. Now, this question is asking you to think back about why enzymes are actually important. Now, as we said before, enzymes are biological catalysts, which means that all of us have enzymes present within us. Now, the reason why we have enzymes, we know, is because the processes in our body naturally takes place too slow all right, to maintain life. So if we did not have enzymes present within us, then the processes would not take fast enough for us to maintain life, and we'll all, unfortunately, pass away. Now, enzymes help us to speed up this rate of reaction, all right, to carry out different metabolic processes. So one reason why organisms need to maintain a constant internal temperature is tied to enzymes speeding up reaction and also to their optimum temperature. So as the graph showed us, each enzyme has their optimum temperature, and if it is that our internal temperature goes above a particular temperature, this can affect the optimum temperature for a lot of enzymes and cause them to become denatured, which will result in a slowing of your different metabolic processes. Now, what are 2 precautions that the students could or should take when doing this experiment? So there are 2 things to highlight, or we should have realized from the question. One was that they were given their potato samples to crush. So one precaution would have been to crush the potato in a uniform manner, or ensure that there was uniform crushing of the potato, because crushing the potato helps to increase the surface area, and you do not want an ununiform crushing of the potato where some or a particular section or some potatoes in certain test tube are crushed uniformly, evenly, and there's a large surface area where others were not crushed uniformly and it has a smaller surface area, so the reaction will be different because of that error. Another precaution was that they said that the different water baths were kept at a constant temperature. So the students would have had to be maintaining or watching the different thermometers which would have been included in the apparatus setup to maintain that particular temperature. So they would have been watching those thermometers to ensure that it doesn't go above or below that constant temperature that they should have been maintained. All right, now it's asking you, how could you have modified this experiment to investigate the effects of pH on enzyme activity, which is normally a common question, or is normally a common lab report that they would like you to do, where you investigate the effect of temperature and investigate the effect of pH on enzyme activity. So you could have modified the experiment by keeping the temperature constant, since the experiment before would have highlighted that 40 degrees Celsius is the optimum temperature for your potato catalase. You could have kept it at that constant 40 degrees Celsius, and then you would have had 4 test tubes of that potato sample, and each test tube would have been given a different pH. So instead of you alternating the temperatures, you are now alternating the pH, and that will help you to investigate if pH has an effect on enzyme activity. B asks us, name 2 enzymes that function in protein digestion and describe the role of each. All right, so 2 enzymes that function in protein digestion, we have pepsin, which we know is supposed to be breaking down our large polypeptides found in, our large proteins into our polypeptides, and it's found in our stomach. And then we have our trypsin, which is found in our small intestine, and it is normally secreted by our pancreas, and it continues to work on the protein, but what it does is that it breaks down the protein into smaller peptides. Now, the question naturally moves to talk about digestion. So it says, before food can be chemically digested by enzymes, it must first be mechanically broken down in the alimentary canal. So it says, name 2 parts of the alimentary canal that are responsible for the mechanical digestion of food. So 2 locations that we have is our mouth And the structure in our mouth, which helps with mechanical digestion, would be our teeth. And then the other location will be our stomach, while the structure in our stomach, which helps with mechanical digestion, will be the walls of the stomach, which helps with the churning process. Then it's asked us to give 2 reasons why mechanical digestion of food is important. The first reason has to do with when we are eating and about to swallow our food, where we help to break up our large food into smaller pieces that can be easily digested. All right, as well. Well, the teeth helps with breaking the food into smaller pieces, while our saliva and our tongue helps with softening and moistening our food to roll it into a ball so that we can easily swallow. And then the other importance of mechanical digestion is to help to increase the surface area for when chemical digestion is going to take place, so we can efficiently or effectively break down our complex food molecules. Right, and this is the last question, or the last part of the question on the enzymes, where we're going to explain the significance of chemical digestion of food. Now, apart from our foods just being larger in structure, they also are composed of complex molecules. And chemical digestion helps us to move our complex molecules, or to convert our complex molecules into easier, more absorbable, simple molecules. So the main purpose of chemical digestion, as I stated, is to convert from your large complex molecules into your smaller, easily absorbable molecules that our body can actually use. Right, so at this time we will start talking about genetics and inheritance. All right, so we're going to start talking off about anemia. All right, so it says, well, A says, state 2 causes of anemia and describe 2 signs/symptoms of this disease. All right, now, anemia can be caused by inherited red blood cell disorder, all right, which would be like a genetic mutation to your red blood cell, which changes the shape of your red blood cell. Another cause of anemia is deficiency, where you can have iron deficiency anemia. You can have pernicious anemia, which has to do with B12 deficiency, and you can also have a folate deficiency to cause anemia. And then you have your gynecological reasons why you can have anemia, which means that there are certain females that have a heavy menstrual cycle, which can result in them having anemia as a result, or as a byproduct. Now, your signs and symptoms of anemia, you have dizziness, headaches, fatigue, you will have pale skin, and you often have cold hands or feet. All right, so the next part, or B, says sickle cell anemia is caused by the presence of a recessive allele. Persons who are heterozygous and carry the sickle cell anemia are more resistant to a type of severe anemia than persons who are homozygous for the normal hemoglobin allele. Now, I asked us to state what is meant by each of the following terms: homozygous and heterozygous. Homozygous means that the person or the gene tends to have 2 identical alleles, 2 alleles that are the same. Or heterozygous tells you that the gene will have 2 alleles which are different. Normally you have a dominant and a recessive allele. Now, the next question is asking us to actually draw our Punnett square. All right, so it says, with the aid of a genetic diagram, draw how a man and a woman who do not have sickle cell anemia can have a child who has the disease. Now, it's a 6 marks question, which means that it's going to ask you to have the genotype of the parent. All right, you're going to have to indicate the genotype of the mother, the genotype of the father. You're going to have to indicate or draw a Punnett square and fill in the data or the possibility of the offspring. And then you're going to have to write the results that you could possibly get. Need the table. Table. Oh, hold on. Let's move my cursor. I forgot about this question. All right. Okay, so first we're going to write the different symbols and what they mean. So for this question, we're using S. So we'll use capital S to indicate that the person has a normal red blood cell or normal shaped red blood cell. And then we'll have a common S to indicate that they have a sickled shaped red blood cell. So since the question asks you how it is that a parent who the mother and the father do not have the disorder, but they are able to have a child with the disorder, this is indicating to us that the parents would be carrier of the gene. So we would write the mother's genotype and the father's genotype. And then we'll now drop our Punnett square. All right, so we'll write our father's allele at the top and our mother's allele to the side. Now, all we're going to be doing in this section where we are writing the possibilities of offspring is to just essentially organize the alleles. So we have a capital S from the father and a capital S from the mother. So we write both of them right here. We have a common S from the father and a capital S from the mother. We have a capital S from the father and a common S from the mother. And then we have a common S from the father and a common S from the mother. All right, so now the last step of it is to write what our probability for the offspring is. So we have it represented here genetically, but we have to write it out for the persons who will be marking the examination. So each box is supposed to indicate a quarter, which is essentially 25% out of 100. So for our capital S, parents who are carrier for the disease has a 25% chance of having a child who has normal, a child who has a regular shaped red blood cell. since we are seeing heterozygous, meaning a capital S and a common S in two different squares, we would have our two 25% adding together to give us 50%. So the parents who are carriers have a 50% chance of getting a child who is a carrier for the sickle cell trait. And then we realize in 1/4, we actually have two common S's. So the parents will actually have a 25% chance of having a child who has sickle cell anemia. All right, so the question is essentially asking you to do all of this where you identify what letters or what symbols you are going to be using, highlight the parent's genotype, work out they said a graphical device or a diagram, which will be your Punnett square, work it out, and then tell them what is the meaning of the possibility or the probability of offspring that you have. Okay, so at this time we'll continue where the last part of the question says, explain why the sickle cell allele is more common in the population in countries where the severe form of malaria is present. All right, so essentially when you're learning this topic, they normally like to use sickle cell anemia as an example, and they tend to show you data which shows that sickle cell anemia is actually more prevalent in countries, well, in African countries or in persons of African descent. All right. Now, the reason for this is because malaria is also present in those countries. Now, what scientists have found is that we do know our sickle cell anemia is a genetic mutation or as a result of a genetic mutation. They realize that persons who are carriers for the sickle cell allele are more resistant to developing or actually getting the severe form of malaria. All right, so as we know about selection pressure or variation or about natural selection is that whenever there is an advantageous gene present within a population, the likelihood of that gene being present increases because it will be passed down more frequently from mother to child. All right? Now, since sickle cell anemia gives this advantageous edge to persons who are living in countries who has a severe form of malaria present, you tend to see a higher percentage of it in the population. So you will see parents who are carriers for that disease or for that trait. And when these parents are carriers for that trait and they copulate together, they tend to have, as you saw on the board, a 50% chance of having a child who's a carrier. Or if they do have that 25% chance of having a child who has a sickle cell trait, that child tends to copulate with someone else, which results in them making a child who is a carrier for the trait. So it's rooted in the fact that the sickle cell trait gives a genetic advantage where you're more resistant to malaria. And as a result, since it's viewed as advantageous, it's found more frequently in the population. So the next topic that we're going to be moving on to is movement. And we know that we have movement in animals and we have movement in plants, right? Now, we'll be looking at our question, which says, in an experiment, some students put corn seeds into each of 2 test tubes labeled A and B as shown in the figure on screen. Now, it says, write a suitable aim for this experiment. Now, in order to write this aim, you would have to look very carefully at the image. So we realize essentially that the 2 test tubes seem to be similar. The plug or the stopper for both test tubes are similar. Both of them have moist cotton wool, and the only difference that we can see is the temperature for each test tube. So a suitable aim for this experiment is to investigate if temperature affects the rate of germination in seeds. Okay? All right, now it says, in which of the test tubes, A or B, would germination more likely occur? And it says, give one reason for your answer. Now, in order to answer this question, you would have to go back or try to recollect your knowledge of enzymes, which we did previously. Now, remember when we were talking about enzymes, we said that you would have optimum temperatures for different enzymes where they work the best at. Now, that is the basis on which this question, or you will get the answer for this question. So I'm going to just go back to the figure that was on screen where we saw 20 degrees Celsius and 10 degrees Celsius. Now, we would be choosing test tube A to indicate where we think germination would likely occur. And this is because there is an increase in temperature and it's closer to the room temperature where we know enzymes work the best. All right, so at 20 degrees Celsius, we would expect that there would be more enzymes being active and actually breaking things down to allow the plant or the seed to germinate and start to grow. So it says, state 2 roles that moisture from the cottonwood plays in the process of germination. So this question essentially is based in your knowledge or what you would remember about talking or defining or identifying the different parts of a seed and their function or their role. So we know that for our seeds or dicotyledonous seeds, we have 2 cotyledons. When we have our little pea seeds on our red beans that we like to make rice and peas with, all right, if we happen to soak them and some of them split, we see 2 little white things, all right, those are our cotyledons. So in those cotyledons are dehydrated enzymes. So the moisture from the cotton wool is essentially activating the enzymes in those seeds or in those cotyledons, which will allow a lot of metabolic processes to take place to make the seed grow. Another role is to help with the softening of the tessa, which is a seed coat, meaning like when you're going to put your peas to soak, those little red coating on your red peas, those are the tessa. So it helps with softening the seed coat, which allows when germination has occurred and the plant is growing, the radicle or what is going to be the root system of the plant is able to easily penetrate the seed coat to actually anchor the seedling in the ground. So those are the 2 main reasons or the 2 main roles of moisture in our cotton wool. Now, on screen, you're seeing another figure, which is another little experiment that was set up. And it asks you what type of movement is shown by the plant in figure 2. Now, we know that plants have 2 types of movement. They can have nastic movements, or they can have tropism. And we know an example of nastic movement is, as we Jamaicans, a plant that we Jamaicans know, which would be shame old lady or touch me not, when you touch them and they immediately close. All right, that is an example of nastic movement, meaning it's reversible. And then we have tropism, which is where your plants will be growing towards a particular stimulus. So the type of movement that we're seeing on screen is tropism, and the stimulus is light. So to be even more specific, we are seeing phototropism on screen. The next question says, state a hypothesis that the students could test by using the apparatus and materials shown in figure 2. Now we know that our hypothesis, I'm going to go back to the figure, or to figure 2, where we saw the apparatus and materials that were set up. All right? Now, we know that our hypothesis is supposed to essentially be an educated guess based on the information that we have or the knowledge that we would have had. So our hypothesis could be that our light source will affect the direction that our plants will grow, or our light source will cause our plants to grow towards, or our plants will grow towards our light source. All right? It says the students want the seedling to grow towards the ceiling. Suggest how they could manipulate the apparatus to get these results. So once again, I'm going to go back to the figure. So we see that in the figure, there is a hole cut on the right side of the box where the lamp is. If it is that the students want the seeds or the seedlings to actually grow towards the ceiling, what they will have to do is that they will have to close up first the hole that is towards the right side of the box. They will have to cut a hole on top of the box directly above the plant, and then they would actually have to move the desk lamp or the light source to be placed above that hole, and that will stimulate or encourage the plant to grow towards the lamp. All right, then we're going to move on to cells. All right, so this is a topic that we would have been introduced to from grade 7, and we keep revisiting multiple times, or we would have learned about it at different journeys along our life. All right, so it's showing you a typical plant cell or an animal cell, and it's identifying certain structures and asking you to name them. Now we have the first structure named, which is A, or identified, which is A, and we realize that this structure is found in both the plant and the animal cell, and since it is identified as the outer layer, the outer lining for your animal cell, we know that this is our cell membrane. Then we have B, which we see is only found in the plant cell, and it is before the outer layer of the cell membrane, so we can identify this as our cell wall. Then we see C, that is found in both cells, and we would identify this as our cytoplasm. Then we have D, and based on the structure identified in the plant cell where it's large and it takes up majority of the plant cell, we know that this will be our vacuole, and we know that our animal cells tend to have small temporary vacuoles, while our plant cell has large permanent vacuoles. So the next question says, state the function of any three of the structures identified in AI above. So I'm going to go back to the image on screen, or to the plant cells, the plant and animal cells, and we're going to just state the function of all four of these structures. So first we have our cell membrane, which is A, and we know that our cell membrane by design or by structure is supposed to be selectively permeable, which means it only allows certain substance to enter the cell. So the main purpose or the main role of your cell membrane is essentially to act as a filter or as a guard, which only allows in certain substances based on its composition or charge into the cell. Then we have B, which is our cell wall, where we will have it helping with the structure or the shape of the cell to give it that fixed shape, and it also helps with preventing the cell from bursting. All right. The next thing we have is our cytoplasm. Now, our cytoplasm is a jelly-like structure where we will have our organelles found within. So it essentially holds the different organelles in the cell, and it also has different enzymes found within it, which helps to carry out different metabolic processes. The last thing we have is our vacuole, which acts as a storage unit, essentially, where it either stores water, it can store different food, and it can also help with temporarily storing your waste product. All right, then we have another question which tells us a student collected 2 specimens, A and B, from a seashore during her biology field trip. She used a microscope to determine which structures were present in the specimens and found that specimen A contained more mitochondria than specimen B. Suggest 2 reasons why specimen A had more mitochondria than specimen B. All right, so the 2 reasons that we have is high energy demand for its function and metabolic activity and synthesis. So for your high energy demand for its function, the cells would normally be performing more physical demanding work, which means that they need a lot of energy to essentially function or to carry out their different activities, which will mean that they need to have a lot of mitochondria, since we know that mitochondria is where respiration would have taken place to produce our cellular energy, ATP. The other reason is metabolic activity and synthesis. All right, so you have certain cells or cell processes that produces, or you have certain cells that process a large quantity of nutrients, or they normally synthesize or make complex molecules. And since they are carrying out these different processes, they also have high energy demands or needs. And as a result, they will have more mitochondria present within their structure to carry out or to do these different activities. All right, so the last part of our cell question says, using 2 named examples, explain why cell specialization is important for cells to function effectively. So 2 reasons are 2 important. It's for efficiency and division of labor, which is something that we normally know or we tend to think of. And the second one is for greater complexity and organism survival. Now, for your efficiency and division of labor, if it is that we did not have cell specialization, then we would have all of the different cells in our body carrying out all of the different activities, and this will result in these cells taking a lot of time to do these different processes and to also do them poorly. So cell specialization actually allows the cells to develop certain unique structures that will allow them to carry out their job a lot more efficiently as they are focused on that particular task. So they're doing it extremely well. And an example of that is our red blood cells, where the shape for oxygen, the shape for a red blood cell, where it actually lacks a nucleus so that it can carry a lot more oxygen present or within its molecule. Then the other one is the greater complexity and organism survival. All right, so this division of labor enables the creation of our tissues and our organs to work together to actually be able to allow the organisms to grow larger and carry out more complex actions and adapt to different or diverse environments, where we'll have our different, like our tissues, our muscle tissues, we'll have our organs being produced like our heart, all right, which all helps with working together, working with the lungs to be able to carry out different processes so that we can actually be able to survive in adverse or different environments. All right. So our next topic is classification and variation. So on screen, we're seeing a figure, or figure 3, all right, which is, it is supposed to be showing the different breakdown of living organisms with the different types, also with some different features or examples present for each. Now, the first question, as we will see, is going to ask us to complete the classification by writing the correct answers at the different parts identified. So for I, which would be where we have types of cell, and we have eukaryotes identified already, it's asking us to identify the other type, which would be prokaryotes. And we saw it gave an example with kingdom, Monera, and an example of it is bacteria. Then under eukaryotes, where we see number of cells, we see one division says unicellular, and the other division is empty, which is II. So we're asked to fill this in, which would be multicellular. Then if we continue under unicellular, we see it, or we would have seen it give the kingdom Protista, and then it asks for an example which we are supposed to fill in, which would be protozoa. So that is III would be protozoa. Then if we go back over to what we had identified at II to be multicellular, we see presence of cell wall, yes and no. For no, we see, which is V, it asks for the kingdom, and it gave you an example, which is fish, and we know that kingdom would have been animal or animalia. Then, if we go to type of nutrition, it says autotroph or heterotroph. And for heterotroph, we see it identify kingdom, fungi, example is yeast. And for autotroph, it asks for the kingdom, which would have been plant or plantae. All right, so just filling in those missing words would have allowed you, as you will now see, to get your 5 marks. Now, B says each kingdom is further subdivided into taxas, with the lowest rank being the level of species. Now you're asked to define the term species, and it's asking for a biological definition or your biological concept definition of species. So we can define species as a group of living organisms which would consist of similar individuals who are able to interbreed together and produce fertile offspring. So in your definition of species, you have to indicate that they're able to not just interbreed together, but produce fertile offsprings. The next part of the question says, using the human species as an example, explain why physical appearance or morphology is not always useful for identifying organisms belonging to the same species. All right, now physical appearance has to do with, if we're looking at the human species, it has to do with hair type, their complexion, their height, what we might look as their silhouette or their shape. And what it's saying is that it's not always useful to identify organism belonging to the species based on those characteristics. Looking at humans, we can see that there are varying complexions that persons may have, varying hair types, all right, varying eye colors, varying foot size, varying heights, and yet still all of these organisms are still able to interbreed together and produce fertile offsprings, which tells us that they are the same species. So the physical appearance cannot be something that you use to classify a species. All right. Now, the next question says, suggest one reason why biologists use the biological concept of species instead of physical characteristics to classify organisms, which is tied essentially to the question above, because physical characteristics can be misleading. Oftentimes, which we tend to see in our birds or reptile species, where the animal may look similar, but they're of two different species, meaning they are not able to interbreed together. So they can't copulate at all because they may have different mating styles, or they may have different sexual reproductive structures. And if they are even able to copulate, they are not able to produce fertile offspring. Fertile offspring means that their child should also be able to produce an offspring as well. Okay? So that is one reason why they will normally focus on, when they're defining species, this species has to be able to copulate, the male and the female species come together to produce a child that can also produce a fertile child as well. Then we have C, which says some lizards were washed away on a log during a hurricane and landed on a remote island. Several years later, they were classified as a new species. I says, using the biological concept of a species, state two reasons why they were reclassified. Now, these have to do with the two different things you would have identified from the biological concept of a species, meaning, are they able to interbreed, and are they able to produce fertile offspring? So would have had an original species of lizards that of course could have interbreed normally and produce fertile offspring, and then we had some that were actually washed away to a remote island. Since they're at a remote island, they are away from the original set, which means that over the period of 7 years, they would have changed certain features, have certain mutations in order to adapt on that island, which could have affected their ability to now be able to interbreed with the original set. Or if they are able to, by some chance, interbreed with the original set, the child that they produce would not be fertile, or would not be able to produce fertile offspring. So that meaning that child would not be able to produce a child of their own. Then it says, the new species of lizard living in the mountainous region of the island were found to be smaller than those living on the lowlands. Suggest two reasons for this variation. One reason could have been the adaptation that the lizards will now have to do because of their environment. All right, they would now have to be able to can effectively or efficiently hide from their predators, which means that they would have to change themselves dramatically in size, all right, to be able to can fit in certain crevices and hide behind certain trees, rocks, all right. Another reason could also be that perhaps there was a diet change based on the availability of food that they would have, all right? So from an energy efficiency point of view, having a larger body means that they would now have to get a large amount of meals or a large amount of organisms to consume, which means that the amount of prey that they have could have decreased in size. So over the years, it could have changed inside or reduced in size to match the amount of available prey that is present. Then we're going to move on to our last topic, which is sexual reproduction. All right? So the first thing we have on screen is that, well, it's a diagram showing the changes occurring in Anna's uterine lining. So it says Anna has a 28-day menstrual cycle, and figure 3 shows the changes which occurs in Anna's uterine lining during the menstrual cycle. Now it says name 2 sex hormones which are associated with the menstrual cycle. And the two hormones that we would have had is estrogen and progesterone. All right, now the next question is asking us to sketch a line for each hormone, which shows the levels of the sex hormone that we would have listed above. Now, normally when we are sketching the line, we would have identified day number 14 to be the day in which we would have had ovulation taking place. All right? So for our estrogen, we would have had a slowly increase in a line graph, and then we would have had it peaking over day 14, and after day 14, it would have gradually reduced or dropped drastically. All right? And then for progesterone, we would have had it gradually increasing past day 14, and then it would have elevated or increased in height, and then it should drop closer to day 27, day 28. Okay? So just to repeat, because we wouldn't have the line graph on screen, for your estrogen, it would normally peak before day 14. So if you're doing a line graph, you would have had the line steadily increasing and then peaking right before day 14, and then it drops drastically and go back down until it reaches to day 28. And then for your progesterone, it would have slowly been increasing, but it wouldn't start to peak until after day 14, where it is peaking about around day 21, 22, and then it will decline drastically. All right, so B says, Anna recently became pregnant. Explain the effect that pregnancy will have on the two hormones that we identified, which is estrogen and progesterone. Now, the first thing that you should note is that pregnancy is going to cause estrogen and progesterone to increase in production. Now, the reason why we'll have estrogen increasing in production is because estrogen helps with developing our fetal organs. It helps with stimulating the growth of the maternal breast tissue and also increases blood flow, which is why persons tend to say that persons who are pregnant have this natural glow about them. It is actually estrogen that gave them that maternal glow. All right? And then estrogen also helps to promote growth and function of the placenta, and we know that the placenta is important to act as gaseous exchange for the fetus and the mother, and to provide the child with nourishment, or the fetus with nourishment. Then we have progesterone, and we'll just list two reasons why it's important, or the effect that it has on the pregnancy, where this hormone helps to maintain the thickened uterus lining, and it also helps with thickening the cervical mucus to block infection. So we know at the cervix, there's supposed to be a little mucus that helps to prevent any bacteria from essentially entering into the uterus. So it helps to thicken it to ensure you have that mucus plug present. And as I said, it helps with maintaining or thicken the uterus lining for the developing fetus. All right, so it says, after the delivery of the healthy baby boy, Anna decides that she does not want to have any other children at this time. She, however, wishes to have another child in the future. It says, recommend one suitable birth control method which Anna could use to prevent pregnancy. A suitable birth control method would have been her IUD. All right? She could also go on the pills or on the daily pills, which are get an injection which some mothers get every 3 months. All right. State two advantages and one disadvantages of the birth control method recommended above. All right. So for the IUD, it is normally someone you normally go to the doctor for them to insert it, and it stays in place. All right, so it normally stays in place, and you have different time frames or different types that you can get, and you have also different time frames in which it can be placed within you. You can have a 5-year period, or you can also have a 10-year period. So it is accommodating in a sense based on the future plans of the mother and when she decides to get another child. All right. A disadvantage is that for the IUD, it is something that the doctor tends to have to insert and remove, which some persons or some females may view as an inconvenience, because they would normally have to schedule an appointment to have it actually removed. All right, and then the last slide says, not all birth control methods can prevent pregnancy. All right, not all birth control methods can prevent both pregnancy and infections from sexually transmitted diseases. It says state 2 types of sexually transmitted diseases. All right, we know about HIV, we know about AIDS, we know about gonorrhea, we know about syphilis. All right, so unfortunately, there are many types of sexually transmitted diseases. It says identify a birth control method which will be effective at preventing pregnancy and infection from sexually transmitted disease. Infections which we know would be our condom. You can have the male condom or you have your femidom. All right. We want to shout out to @Shanti on YouTube, who was very responsive and answered each question correctly. All right. I hope this entire presentation will be helpful for all of our candidates. We wish you the very best, and thank you very much to all our CSEC biology candidates. Bye.